at the University of Windsor, but most importantly, I'm the director of FishCAST. FishCAST is an NSERC-funded graduate training program where we're training the next generation of leading scientists in freshwater fish conservation and management. And just before we begin, I wanted to know where everyone was coming from. I myself am coming from the beautiful territory of the Three Fires Confederacy of First Nations, and that includes the Ojibwa, the Odawa, and the Potawatomi peoples. FishCAST is a community. Well, we're actually working with our trainees, our graduate students, to try and move beyond land acknowledgements and to really meaningfully engage and to find ways to meaningfully engage and to acknowledge and to build relationships that really do acknowledge the history and the connection to and the knowledge on the land in which we're situated, the lands in where we're doing our research. And it's kind of like a common theme that we're trying to do our best to weave throughout in terms of what we're exposing our trainees to and the research opportunities they have and just the people that they meet. Our other goal is to really address the systemic barriers that are specific to our fields in freshwater fisheries, conservation and management. And we're trying to work collaboratively with others and with allies on finding ways and people to work with, right? And trying to overcome them. It's a long process, obviously. We're starting and we're really excited to see where it takes us. And the reason that's really motivating us is that we are and we will all continue to be treaty land inhabitants. And from there, I want to welcome you all to our FishCAST seminar series, where today we'll be listening to an exciting seminar presentation by Dr. Brian Dixon. Brian Dixon was born in a castle in northern England. Now, I've seen pictures. I haven't seen pictures of his birth, but I have seen pictures of this castle from Whitehaven. And what I think we all need to know that it was repurposed as a hospital afterwards. I think he was actually born in the hospital that was previously a castle, because he doesn't make claims to any royalty whatsoever. But he then moved to Canada, and that's where he received his bachelor's degree from Wilfrid Laurier University, and he received his master's degree from the University of Guelph. He moved over into the Maritimes to pursue his PhD thesis at Dalhousie University at Halifax, Nova Scotia, where he examined the immune responses to parasites, not just of fish, but of seals as well. Following his successful dissertation, he received an NSERC postdoctoral fellowship to go to Wageningen University in the Netherlands. And then he also received a fellowship from the Medical Research Council of Canada. And he actually went to Stanford University in California. We were lucky enough for him to come back to Canada, where he's currently a professor in biology at the University of Waterloo. And Dr. Dixon has had such an incredible impact in his field, and his expertise in his field has awarded him a Premier's Research Excellence Award from the province of Ontario. He's a Canada Research Chair in Fish and Environmental Immunology. And in 2014, he was awarded the NSERC's Canada Synergy Award for his collaboration with the aquaculture industry. So he does work with a lot of partnerships and communities. But what does Dr. Dixon actually do? Well, he's internationally recognized for his work that focuses on fish immunity and the influence of environmental factors such as temperature on fish immune systems. He's an expert in the development of diagnostic assays for assessing fish health status, antimicrobial peptides, and applying fish immunity and immunology research to improve aquaculture production worldwide. He's quite the globetrotter. I was able to eke out of him that he's not only worked across Canada, but he has work that he does in participation with governments and industry in Chile, Cuba, Spain, Panama, Argentina, and Australia. Dr. Dixon's also a member of GenFish, and if you were here last week with us, so was Dr. Margaret Docker, and that is, GenFish is an acronym, and it stands for the Genomic Network of Fish Identification, Stress, and Health. And this is a networked team of researchers, but not just researchers, but professionals and community members who are working to develop and validate genetic tools to be able to cost effectively assess basically the health, location, and abundance of Canada's freshwater fishes. But we're really proud to claim Dr. Dixon as one of our own in terms of being an integral member of the FishCAST community. Interestingly enough, though, Dr. Dixon did a quick pivot back when the pandemic first started, and he pivoted his lab to COVID-19 research, where he developed a test for the SARS-CoV-2 antibodies in blood. And then he's currently using to research the immune responses to the viruses in patients at local hospitals, but also he has his lab guinea pigs, the student and staff population at the University of Waterloo. But he maintains and he ascertains and he wants to let us know that traveling and studying fish immunity is really near and dear to his heart. And he's going to be showcasing that to us today through his talk on the weird and wonderful world of fish immunity. And if you can please join me in welcoming Brian today, that'd be wonderful. Thank you so much. Brian, welcome. And whenever you're ready, please go right ahead.
Thank you. I'm going to have to try to live up to that. I don't know if I'm going to talk about much travel today, but if you ever catch me and we're having a beer, ask me about being stuck up a river in Guatemala during a hurricane. I'll tell you that one. That's, I don't admit to anything on camera recording this, I guess, but I have bribed customs officials to get into countries. It's just part of the job. All right, let me start sharing here. Let's see, share screen. There we go. All right. I don't know how this is going to work on slideshow. I don't think I can see anyone. Oh, I can sort of see people. I'm doing this, but
perfect.
Okay, you can see my screen?
Looks great.
Okay, good. All right. So yeah, welcome. I'm a fish immunologist. I did dabble in humans a little bit and a little bit in coral and a couple of other things. Primarily, I'm a fish immunologist, and I do want to say that I do prefer it. This is me in my happy place at Yellow Island, which is one of the fish farms we work at, spawning salmon. So that's a Chinook, big mature male. And I do understand that the immune system is sometimes very intimidating for people. So I'll start with a joke. There was a cardiologist and an immunologist at a conference, and they were captured by terrorists. And the terrorists said, "We're gonna ransom you off, but to show we're serious, we're gonna kill one of you. And so the one of you who's contributed most to the world and well-being will live. The other one we're gonna have to kill." So they looked at the cardiologist and they said, "What have you done?" He goes, "Well, I've done all this research. I've developed drugs to prevent people from having heart attacks. I've done lots of life-saving surgeries. I'm pretty important." So then they turn to the immunologist and they say, "What do you have to say for yourself?" And the immunologist said, "Well, the thing about the immune system is it's complicated." And at that point, the cardiologist said, "Just shoot me now." So the immune system is complicated. I'm going to try and simplify things, keep it as simple as possible. A lot of these slides, some of them may have things that look complicated. There's generally a simple take-home message from them there, okay? So, and I've only got about 35 slides. I'm going to answer questions at the end. So again, I'm Brian Dixon. I'm a fish immunologist. I work on the traditional lands of the Anishinaabe, Haudenosaunee, and neutral peoples. I work on the Haldeman Tract, which is the land promised to the Six Nations when they moved to Ontario. But I do work around the world in lots of different lands with lots of different indigenous cultures, and I acknowledge that I work there with their, on their land. So I work on fish immunology, and I wanted to tell you about the weird and wonderful world of fish immunology. If you've ever taken any human immunology, it's not going to apply. And I think that's going to be the lesson I'm going to try and get across today, is if you know anything at all about immunology, it doesn't apply to fish. And there's many reasons for that. So the first thing I want to tell you is how different fish are from humans. The example I'll use is just antibodies. I think we all know enough to know what antibodies are. I'll talk to you a little bit about how different they can be. And then I'll talk about what affects fish immune responses. I'm going to give you some examples. I'm going to talk about some of my older research, but we do have newer research on this as well, about water temperature and how that affects fish immune responses. I'm going to put in some stuff about sex and humans affecting fish immune responses. I've been watching Netflix lately, and I noticed that's what sold, so I figured I'd throw some of that in there. And I have a little bit on evolution, just a little bit, and if that's the part I don't get to, I'll be fine with it. But I'll talk about what evolution has done to the fish immune system. All right. So, and I've got some specific examples of work we've done on all of these. So let me just start with immune systems. And again, very complicated slide, it seems, that looks at all of the immune tissues in all the different groups of vertebrates you can find, starting with the lamprey and then up to mammals. And I think the general take-home message here is as you move from lampreys up to mammals, you get evolutionary innovations, you get new tissues. This is just tissues or organs appearing that help control the immune response. And the immune response is really about control. So in my immunology class, I keep telling them the immune system is the most paranoid system ever, because there's a lot of potential, the way we develop our immune system, there's a lot of potential for autoimmunity. And you really need levels of control to prevent that from happening. So as evolution has gone along, the immune system's got more complicated, really to separate the cells and control them. But you can, oops, sorry, it's a timing thing. You can see already that the fish immune system has less of the tissues and less of the organs compared to, say, a mammal. And so in mammals, all our blood cells develop in our bone marrow. Fish don't even have bone marrow. So they use a little part of their kidney, the front third of their kidney or so, called the head kidney, because it's close to the head. That's where blood cells develop. And the differences go on from there. So already you're looking at a less complex, seemingly less complex immune system, although it's complicated enough on its own. So, and again, there's lots of different components of immune systems. There's like, literally, depending how you define it, tens to maybe 100 different types of cells. There's these molecules called cytokines, which send signals between the cells. There's antibodies, which we'll talk about, which is really just a fancy targeting mechanism to help the immune system figure out what to kill. And then there's proteins on the surface of cells called receptors. Today, I'm really only going to focus on antibodies and receptors. I won't talk about any of the things, but I'd be happy to answer questions about them. But the examples I use will really be just antibodies and receptors. So what about antibodies? What about antibodies in fish? How similar are they to the antibodies that we understand from humans? So I've got a little simplified diagram here of the antibodies that are present in the different groups of animals. And you can see already jawless fishes like hagfish, they don't have what we call immunoglobulins. It turns out, interestingly, they do have things that look a lot like antibodies and can recombine and are specific for binding to pathogens like antibodies, except they're built on a completely different set of proteins. So the immune system that we understand in mammals starts really with the cartilaginous fish, although it's not the only adaptive immune system out there. And you can see, you know, there's different types of antibodies in different groups. In mammals, generally you have IgM and IgD, and we won't talk too much about IgD. The only interesting thing I'll note is that it's not in birds or amphibians. It seems to be conserved in fish. We don't even know what it does in humans very well, but whatever it is, it's important enough that it's evolutionarily conserved in fish. Every organism has IgM. That's the first type of antibody you ever make in an immune response. And then in mammals, you would switch to IgG or IgE or one of these other types as they go along. But you can see fish, really, they have IgM, they have that IgD that we don't know what it does, and they have a sort of equivalent of IgA called IgT. Now, IgM and IgG would be the ones that are normally circulating in your blood. IgA or IgT in the case of fish is really your mucosal surface antibody. So it's the one you would have in your gut or your lungs. But of course, in fish, their skin and their gills are mucosal surfaces, so it's distributed kind of differently. So already you can see that there's different kinds of antibodies in different groups of animals. And I'll show you this. IgT is not even really equivalent to IgA. It plays the same function, but it's a different thing altogether. And just the way the antibodies look, so this is a sort of a rough diagram of how antibodies look. You can see, I showed you before, like you might understand the simple Y-shaped structure of an antibody. You can see it's like 4 proteins in a single unit. But in humans, the IgM, the one that circulates on your blood, is actually 5 of those little subunits linked together into a pentamer, right? And the IgA, the one that's on your mucosal surfaces, is a dimer. I didn't have a similar diagram, but you can see it's 2 of those subunits put together. However, in fish, already you've got IgM is actually a tetramer, and the IgT, the mucosal one, is a tetramer. So they look structurally not like the mammalian ones at all. So very interesting. What's fascinating evolutionary-wise is if you look at the lobe-fin fishes like coelacanths, or fishes that actually led to the amphibians and the tetrapods, the land animals, their IgMs are actually a pentamer like humans. So this tetrameric IgM is an innovation that's only in the bony fishes, not in the main lineage of evolution. So. Also, the way antibody responses happen, I think we all understand that if you look here, it's here's a typical antibody response in terms of the amount, the level, or the amount of antibody in your blood. And when you immunize or you get sick as a mammal, you make the IgM. As I said, you always make the IgM first, and then it sort of plateaus off as you switch and start making the IgG. So you'll get IgG, and then if you get sick again, or if you get boosted with an antibody, you get more of the IgG, and as you get sick or boosted again, you get more of the IgG again. And even the affinity, the tightness of binding of your antibodies, it goes up every time you get infected again. So a second or a third, the affinity goes up. So you can see how it works quite clearly. And vaccination is great. If you vaccinate someone, then when they get sick, they get that second response, and they already have a much higher affinity, much higher antibody level response. It's not how it works in fish at all. I think if you were paying attention to the last one, you could see fish don't even have IgG. So what they have is they have that pentameric Ig or a tetrameric IgM, and then they have just monomers of IgM that float around exactly like IgG. So IgM does both jobs. And what you can see if you look at a fish response, this is a trout, for example, you can see that you do get an increase in the amount of antibody that goes on, and you can see it's still on a logarithmic scale, which is good. The difference is, of course, the timing of this response. If you look at the time scale in the mammals, you can see that the peak of the response is going to be in a couple of weeks, and then when you get a second response, it's in weeks again, so 2 or 3 weeks. For fish, you can see the peak of the antibody response is 15 weeks post-immunization. So their responses do get high in terms of antibody levels, but it takes much longer for it to happen, and then it peaks. And certainly the affinity also goes up, but again, on a scale of 15 to 20 weeks. The difference is, of course, they don't seem to have these secondary responses. If you immunize this fish again or infect them, the antibody titer will not go up any higher than it did the first time. So they don't seem to have the IgG, and they don't seem to have the memory responses whatsoever. So while you're immunizing fish, like you're immunizing humans, you're not really going to get a better response next time. You're just getting a response, and it actually trails off. You can see, a year or two later, you could get the antibody levels still being similar. It tails off really slowly, and there's lots of reasons for that. One of them is that fish don't, at least rainbow trout, don't live as long as humans. Maybe they don't need a memory response, and so they have a different response altogether. And the interesting thing is, I think Dr. Semeniuk said, I work in Panama. I work on cobia. They're warm water fish. Their immune response happens much faster. They will respond in a week or two, and then it'll be over and gone. So it seems to be that this slow response might be just down to the fact that the fish live at these fish, at least rainbow trout, live at a colder water temperature than human body temperature. It may be just temperature. Another thing is, and again, this is a complicated diagram, this is the gene structure of the immunoglobulin genes, the ones that make antibodies. But what I want to point out is, as I said, in mammals, you start with IgM, and then what you do is you actually chop up the DNA and you rearrange this gene in your immunoglobulin producing cells, and then you'll either make IgG or IgA, one of those other types of antibodies. But you'll never make IgM again with that cell, because you've chopped up the DNA and removed that. In fish, what's interesting is I told you that the IgT is not really equivalent to IgA. In fish, it turns out the IgT is actually before the IgM in the gene organization, which is not true in any of the other tetrapods. And what happens here is the cells either produce IgT or IgM, and they do it from the beginning, and then they never change their isotype. So fish immune responses are built on completely different structures of genes, and they don't work the same way at all. Right? So IgT is in a weird place. It's the only vertebrate where this happens. So you can see, although superficially, and this is again the theme of the day, although superficially it looks like they have the same types of antibodies or similar types, you can't count on them playing the same role or the same function. Fish don't have IgG. The IgM plays that role as well. The IgT is present on mucosal surfaces like IgA, but its regulation, the way that the genes produce it is all completely different in fish, and bony fish anyway. Okay, so that's antibodies, just to show you how weird and wonderful they are. I did want to talk a little bit about temperature, because I think, as I pointed out, temperature does affect immune responses. And I'm going to talk a little about some receptors. These are some of my favorite molecules in the immune system. Again, it looks complicated, but it's really a fascinating, simple structure. And I'll talk about how water temperature affects the way immune responses start or don't in fish. So the molecules that I really like are called major histocompatibility complex, or MHC receptors. There's various different versions here, but let's just look at the simplest version. They sit in the membrane of your cells, right? And although there's two different types that deal with different types of pathogens, the MHC class 2 deal generally with bacteria that are floating around outside your cells. The MHC class 1 deals with things like viruses that get inside your cells. And they have different polypeptide structures. But what you can see is they have basically the same protein structure. And what they have is they're stuck in the membrane of your cells, and they have at the top this little, what they call peptide binding cleft. So what they do is very interesting. They grab onto a small fragment, a small amino acid chunk of the pathogen, and they go up to the surface of your cells, and they present it to your immune system cells. And your immune system cells can see what's in that little groove, and if it's something that looks like it's a threat, they start immune responses. So these receptors are what start your body making antibodies or responding in any serious way to a pathogen. So they're critical in starting immune responses. Today, I'm going to talk to you mostly about these MHC class 1 ones that you can see. There's two polypeptides there. There's one here, the big one that has this alpha 1, alpha 2, and alpha 3 domain. We generally call that the heavy chain or the big chain. Oh, sorry, I've got this on a timer. I should not have done that. And then there's a smaller polypeptide piece called beta 2 microglobulin, right? And you need both of those pieces of protein together to make the receptor to bind this peptide and present stuff to the immune system. Now, what I'm going to show you is that this is the expression of these proteins is intimately linked to temperature, right? So when I was a PhD student, there was a paper that came out on humans that said if you grew human tissue culture cells at 27 degrees Celsius, these proteins were all messed up. The beta 2 microglobulin piece didn't come out on the surface. These parts of the protein came out, but they didn't have the peptide, and it did not work very well. Now, if you're a human being and your body temperature is 27 degrees Celsius, you've got bigger problems than being infected with a virus. But what I thought is fish live at a wide range of temperatures, you know, and it would be ridiculous for them to mess up the expression of these genes at low temperatures because they have to live there and they'll get exposed to viruses. So I decided to set up an experiment to show that these proteins are expressed on the surface of fish cells at all temperatures because that's where they live. So the experiment I set up, and this was done many years ago when I lived in Wageningen, Dr. Semianek, there'll be a pronunciation test for that a little later. And what we worked with was carp. So we had carp, and what we did is we took some of the carp, and we took we started with 18 fish. We kept 6 of them at 24 degrees Celsius, which is their normal culture temperature. So we keep them all along. We dropped some of them down to 12 degrees Celsius, and then because you can't drop them all the way to 6 right away, it's too much shock, we took 6 of those and dropped them down to 6 degrees Celsius. So at the end, we had 3 groups of fish. Fish that stayed at 24 degrees Celsius, fish that were dropped to 12, and fish that were dropped to 6 degrees. And then we kept them there for a few days, and then we moved them back to 24 degrees in the end, all of these fish. And we looked at the presence or absence of those proteins I told you about, MHC class 1, on the surface of their cells, all right? So a technique that we used is called flow cytometry, which is a very, very cool technique in which you can take a single cell, run it through a little microfluidic device, and see what's happening with that cell. So this looks like, again, like a complicated graph, but it's not really that complicated at all. On the y-axis here, you just have the number of cells that you're looking at. How many cells are in each of these categories? And along the bottom, we've got its fluorescence intensity. We tag the cells with a fluorescent marker that sticks to a specific protein. And so you can see, for example, here, this is the carp immunoglobulin. So this is looking at their B cells, their antibody-producing cells, and do they have antibody molecules on the surface of their cells? And I'm only showing you the 6-degree data, but you can see fish at 24 degrees Celsius. If you look at their blood cells, they have some cells that do not have immunoglobulin, because not every cell in the blood is a B cell, and you have some that have immunoglobulin. So they're fluorescing when we stick a tag on there to show us that they're B cells, or they have immunoglobulin on the surface. And you can see that no matter what we did with the temperature, there was always, 25 to 30, 40% of the cells had immunoglobulin on the surface. So no matter what happens to the temperature, the fish cells had antibodies on their surface. However, when you look at the two parts of the protein I was telling you about, there's the beta-2 microglobulin, and that's just our shorthand for that other chain, the heavy chain. You can see that many of the cells, or most of the cells in the blood, have both of these proteins on their surface when they're at 24 degrees Celsius. When we got them to 6 degrees Celsius, after 3 days at 6 degrees Celsius, this protein started to disappear from the surface of their cells. Most of the cells now don't have any protein there. And by 6 days at 6 degrees Celsius, these proteins had completely disappeared from the surface of the cells. However, when you put them back up to 12 degrees Celsius, these proteins came back up on the surface of their cells. So my hypothesis was that fish would have these proteins on the surface of their cells at all temperatures, because they could get infected at all temperatures. And to quote T.H. Huxley, that is the great tragedy of science, the slaying of a beautiful hypothesis by an ugly fact. It turns out they do turn off the expression of these genes, and they are not presenting any of the peptides to their immune system. They're not initiating their immune system at low temperatures. Now, these fish that we kept at 6 degrees, they're carp, it's not lethal for them, but it's the bottom end of their temperature range. It's probably the temperature of ponds they'd be living in over winter. So it seems like they're not eating very much. They're sort of like going a little bit lethargic. So maybe they're not eating. They're a little bit temperature stressed. They're low on energy, and they're shutting down their immune response to save on energy, because immune responses are energetic demanding. And it turns out, even in hibernating mammals like squirrels, you can inject them with something and they won't make antibodies at when they're hibernating either. So there's something fascinating going on here where they don't make immune responses at low temperatures. Now, that's carp at 6 degrees Celsius, and they're shutting down the part of the immune system that responds to viruses. When I came back to Canada, I wanted to work on salmonids, so I started working on rainbow trout, and this is a different way of looking at it. This is called a Western blot, but each of these little bands represents a protein, one of the proteins that I can detect on the blood cells of the fish. And this is the other one, the class 2 one, the one that responds to bacteria and viruses. Now, the reason we looked at those is we looked at the ones that respond to viruses like beta-2 microglobulin at low temperature and high temperature in trout, and you can see the proteins there all along. So they do not turn off this protein at low temperature. They keep that one on. But when you look at the protein that responds to bacteria, it turns out one of them, again, just one of the two proteins, they shut it off. There's not very much there. This fish clearly did not read my papers or subscribe to my hypothesis. It has a little bit there. But it turns out that salmonids, it looks like, at the low end of their temperature range. Again, this would not be unusual for a trout living underneath the ice in the winter. They shut down the part of their immune system that responds to bacteria. Now, why the two different fish do that? Maybe it's a reflection of the type of pathogens they face. Maybe trout face more problems from viruses and they want to keep that part of the immune system on, and carp face more problem from bacteria. I don't know. But it's interesting that the two types of fish both turn off their ability to respond, but they do it differently. Okay, and just to show you, I showed you that this beta-2 microglobulin, this virus responding system, the proteins stay present at low temperatures. We subsequently did a little study where we looked at the immune response to viruses at low temperature. This was done in a cell line, and you can ignore that. I mean, this is all the just examples above, but I think you can just see it from the graphs below. This is, again, the ones I looked at in carp, so the MHC class 1 heavy chain, the beta-2 microglobulin. And let's not worry about this other one here for right now. But what you can see is at 14 degrees, if you take the cells and you leave them alone, they have a certain level of the MHC class 1 protein. If you infect them, then they upregulate, they put more of the protein on the surface of their cells. But if you do it at 2 degrees, they don't. Same for that. So even though the proteins that can respond to viruses are present on the surface of the cells at low temperatures, it seems like cell monads don't actually mount an immune response for regulatory reasons. So it turns off that for whatever reason, fish turn off immune responses at low temperature, and not just carp. Some monads do it. I think it's a stress and an energy demand thing, but certainly they don't have immune responses at low temperatures. And that leads you to some interesting questions if you're working with fish. So again, temperature variance suppresses and sometimes delays immune responses because, again, the antibody responses are delayed because they live at low temperatures. It's consistent in many species and cell types, although the specific genes regulated differs. It does cause disease outbreaks. There's lots of diseases in some monads where the low temperatures, I can think of Flavobacterium psychrophilum's 1. There's some fungal pathogens that do this. Certainly for catfish, there was a pathogen that happened when the catfish experience drops in temperature. So as the temperature drops, the immune system suppresses and you get disease outbreaks. So, for husbandry of your fish, this is an interesting problem. And here's one that I always thought is interesting. What temperature should fish be vaccinated at? Because they tend to vaccinate fish when they're putting them out in the net pens. But, depending on the time of year you're doing that, and depending on the water you're using, you could be using spring water for your freshwater fish before you put them out in the ocean in net pens. If your water temperature is like 2 to 6 degrees Celsius, how effective is your vaccination if their immune response isn't really working there at all? So there's a practical question for you. Okay, I'm going to move on now to the second part, and this is humans and sex affecting the immune responses. As I said, I noticed on Netflix it sells, so I'll tell you a little bit about this. The real story here is about implanted telemetry tags. So some of you might be ecologists. You might be interested in tracking your fish. So this was actually a little side project that came up because I was working with an ecologist across the hall called Mike Power. I was on the committee of a student called Ingeborg Mulder, who's from the Netherlands. And she was looking at Arctic char. And, Arctic char are distributed in the north. And there's lots of concerns that climate change might have a bigger impact on them because they live farther north where climate change is going to be a problem. So they were working on fish up in oh gosh, I'm going to lose my mind. Up on the north coast here of Newfoundland and Labrador, and they were looking at Arctic char that migrate out and feed in the ocean off Greenland and then come back. And they wanted to track their movement using acoustic telemetry. So they surgically inserted tags in and released the fish. And this, of course, allows tracking the fish in 3 dimensions. And when they come back into the reservoirs and the river systems, they could track them. All right. Now, Ingeborg, when she was presenting to her committee, was telling us that she lost a lot of tags. So she tagged 56 in the first year and 35 in the second year. And only 22 came back from the first year and 16 came back from the second year. And the thing that caught me was the tags cost $750. So this is a big loss. So I wanted to know why she was losing tagged fish. And she said, well, sometimes they expel them out of the incision, or sometimes expel them just through their body wall cavity. It could be mortality or capture, or it could be susceptibility to disease. And so my question came up, well, what's the impact of the tag on the fish? Is it causing deleterious effects? Is it causing immune responses? Because if you implant certain materials into a body, you're going to have an immune response happening to it. And there was not really much research done on this at the time. How do these tags influence? They're relatively large tags. When you look at these large tags, the size of fish you're putting them in, I think Mike told me the equivalent is putting a small fire extinguisher inside the body of a human being. Could this have something to do with how they're lost, and how does this work? And I mean, most people do monitor fish to see if they survive after they've had the tags in them a little while, but there's no fine scale looking at immune responses. So what we did was we had some fish left over from a study, and they were mature fish, and we'll get to why that's important in a little while. We had some fish left over from a study, and so we thought, well, we'll just take some of the tags and we'll do some surgery. We'll have some fish that are tagged, some fish that get the sham surgery because we want to make sure that the incision doesn't have an effect. And we'll sample the fish at 4 time points. So day 0, just before the surgery, 2 days after the surgery, 2 weeks after the surgery, and 10 weeks after the surgery. Or sorry, 2 days, 15 days, 2 weeks, and then 10 weeks to look at long-term effects. And really, we're trying to do the surgery in the way they did it in field conditions. So we used the same anesthetic, the same surgical tools, everything. In fact, we had Ingeborg do it. So you can see, here's the tags. Ingeborg would make an incision, and you can see just as small as incision as possible to shove this tag in. And you can see the scale of the size of the tag to that fish. And then Ingeborg would stitch them up, and then we'd let them heal. And then we sampled them to see what was happening with the immune response. Now, one thing I did want to note is because these were mature fish, some of them were mature females with eggs. And that became important because that's where the sex part comes in. We had some females and some males, and we were able to differentiate between the two when we did our analysis. But what I did want to point out is these tags stick to the tissues. I don't have all the gruesome pictures here, but you can see they get implanted into the tissues. The fish are clearly responding because they're making a fibrous capsule around them. Some of them had adherences where they stuck to the side of the fish, and some of them actually had little holes wearing in the side of the fish where the tag was rubbing, and you could see where it might have been expelled out. What was interesting was we had one fish out of our study expel a tag. When we cut it open, it didn't have a tag in it, and we never found it. We couldn't even find it in the tanks we were in. So it's interesting that these things get expelled in the wild when we can't even control what's going on in a tank. So just to look and see what was happening with immune responses, we looked at the site of the surgery just to see what was going on there because we wanted to make sure there was no inflammation from the surgery. We looked at the spleen because that's where the central immune responses happen. And then we looked in the peritoneal cavity where the tag was and where it was rolling around. So, I mean, for those of you who aren't immunologists, you don't really need to worry about what these genes are, just that these 3 genes, when we're looking at their expression of them, are genes that cause inflammatory responses. They're the ones that start immune responses, and in humans start fever and all the things that go along with the energetic demands that are required for immune responses. And you can see we had the control fish, the sham surgery fish, and the tagged fish. And you can see at the site of the surgery, there was not really much response. There was some upregulation of one of the genes, but the difference between the control and the tagged ones was not significant at all. So it's not an effect of the tags that's happening. And this may, in fact, be an effect of having the tag in the cavity. And you'll see in a minute there's a response to the tags. So this may be just a localized effect of signaling molecules from the immune system making this happen. But there was no significant difference between the tagged and non-tagged fish. When you look at the spleen, which is central immune responses, it looked like there was some response a couple of days afterwards in the tagged fish, but the statistics said that there was not a significant difference there at all. And you can see, so there's not really much going on in the spleen in terms of central immune responses either. However, when you look at the site of surgery, and I do want to point out the scales are different. Those are the ones that were smaller. This is a scale that goes up to hundreds. You can see in the fish with the tags, there was definitely a significant difference in the inflammatory cytokines 48 hours, so after the surgery, just in the tagged fish, not in the surgical fish. You can see that there was a difference in one of the genes at 2 weeks, and at 10 weeks, you can see that tag's been scraping around inside the fish. And because of the plastic surface, we think that there's a thing called a foreign body response, where antibodies and other molecules deposit on it because of its surface, and then you get an immune response to it. But you can see there is a large response, inflammatory response going on in the peritoneal cavity of the fish where the tags are 10 weeks afterwards, so a chronic response, right? So you can see the tags do have a long-term effect. And again, even if you watch them for a couple of days or weeks afterwards, there'd be less of an effect, but there is a long-term effect that you might not see when you release these fish out into the wild. Now, as I noted before, there's a sex component to this. Because we could see the mature females had eggs, we could differentiate which fish were male and female. And I did want to tell you about what happens in mammals in terms of immune responses and sex. So again, this is a list of autoimmune diseases. And what I'm going to do is I'm just going to point out with and I hate to stereotype, but a blue arrow for the ones where they're more frequent in males, and pink arrows where they're more frequent in females. And you can see most autoimmune diseases are much more frequent in females, in humans and mammals. And this is quite commonly known because our immune cells in mammals have estrogenic receptors. So they will respond to estrogen, and that stimulates them. So in general, women have much stronger immune responses than men, especially when they're pregnant. The downside of this is those stronger immune responses tend to lead to more frequent autoimmune disease, because regulating the immune response and having a well-regulated immune response is what's important for not reacting to self and having autoimmune diseases, as I pointed out. So in mammals, it's the females that have the stronger immune response. However, when we parsed out our immune responses in fish to these tags based on sex, you can see it's actually at the surgical site, there wasn't a difference between the group, the tagged and non-tagged. But when you looked at the surgical site responses between males and females, the males did have a significantly bigger response for some of these pro-inflammatory cytokines than the females. Nothing happening in the spleen, although suggestive of something, but no statistical difference. But in the peritoneal cavity where the tag was, again, almost all of the immune responses we saw when we looked was in the males, and there was almost no response in the females whatsoever. Now, when I looked into this further, there's not really much research done on sex-specific immune responses in fish, because generally we work with immature fish. Certainly for rainbow trout, we work with small trout that are not mature. It's hard to tell what's going on, and you can genetically type them. There's very few papers on this, and the few papers I found suggested that fish immune cells actually have androgenic receptors on their surface, not estrogenic ones. So it seems like it's the males that are going to have the stronger responses, which is what this says. And there is some evidence that females express higher levels of the regulatory cytokines that are molecules that control their immune responses. But what we don't know from this is it's happening in immature females as well as mature females, or are these mature females suppressing their immune responses because they've got eggs in their peritoneal cavity and they don't want an autoimmune response or some kind of response that will destroy the eggs? So there's still lots to tease out about this. Now, we haven't continued the sex part, but because we thought that it was the surface of the eggs that were or surface of the tags that was causing immune response, we decided to try with a couple of different types of tags. So we got some tags from the company that we got them from before Vemco, but they have them coated with a rubbery compound that is supposed to reduce the surface effects. And we got some from a company in Iceland called Antibodies that makes ceramic tags, and the ceramic is supposed to be a little bit smoother. So, and we looked at these, but all three of these tags caused encapsulation equally in the peritoneal cavity. And we haven't completed our analysis, but we have done some of the studies for the site of surgery, which is the muscle. And you can see in each case there was not much difference in the gene expression in the muscle between the surgery and any of the fish where the tags implanted. But when you look in the spleen, it seems like the coated Vemco tags certainly seem to be inducing an even larger immune response. And we have to dig further into this, but it seems like the coating's not helping. So the Antibodies ones the ceramic ones do, at least in this case, seem to be not as much of a response, but we have to dig into this a lot further. Okay. So peritoneal tagging in humans it can affect fish immune responses because they can induce chronic responses. My question is, what about pit tags? They're smaller and they're made out of glass, which is supposed to be immunologically inert, but lots of us put pit tags in our fish, and does that have an effect? It would be nice to know. Sex influences on fish immunity. It's the opposite way in fish than it is in mammals, but it does raise questions. What about all female aquaculture operations? Does that mean that the female fish are responding less well to the vaccination and maybe are not as useful as they could be? What about estrogenic contaminants? So now you're shifting male fish to female fish. Does that affect their immune responses? Is that going to affect their survival at an immune level because of this feminization of the fish? There's lots of questions that could happen there. I think am I at 50 minutes, Tina? Yeah. Okay. I'm going to skip the evolution part. It's about gene duplications. Fish did a lot of gene duplications, but what I can say is the general scheme of fish evolution is that it arose in the ancestors of the vertebrates. The first evidence of the immune system that we use is in sharks, and it goes from teleos up to frogs and everything else. But what I showed you today suggests the fish, because of the genome duplications they didn't talk about, but everything else, do things differently. They've evolved a very different immune system than all of these groups of animals. And if we're going to work with them, we're going to have to understand what's different about their immune system to make sure that we're working with them appropriately, because they're different again from everything else. Anyway, and with that, this is a list of all the companies. It's not everyone from my lab or all my collaborators, but all of the companies that and other universities I've worked with. Thank you very much. I'll answer questions. Let me see if I can stop sharing.
Yeah, thanks so much, Brian. That was really great. I actually learned a lot of stuff. I've got tons of questions, but I'm going to open it up to the audience first. And at this point, we'll turn off recording. So feel